And along the way, the sperm, by the way, is completely incapable of fertilizing that egg. That seems to be a problem right there, doesn't it? Except in the mother, there are chemicals that we say capacitate, make the sperm able to fertilize. So without capacitation, it'd be pretty much the end of life, at least mammals. The sperm arrives, the oviduct is coming down. How do we get the egg to go the opposite direction? If the muscles are bringing the sperm up, how does the egg go the other way? Because let's face it, if the egg just stayed in the oviduct, do you see a problem there? Fertilization could occur. The egg wouldn't go any further, it would implant in the oviduct. And sadly, that happens from time to time. It's called a tubal pregnancy. It's a medical emergency and will kill both mother and child unless something is done. That'd be the end of life right there. If you don't move the fertilized egg down the oviduct into the uterus, you don't have to even worry about what comes next in development, because it isn't going to happen anyway. And in the case of evolution, if you can't get the egg down the oviduct, you aren't ever going to get a second chance to get it down the oviduct, because it's the end of that organism. Boy, talk about irreducible complexity. You know, I don't think bacteria necessarily need a flagellar motor, because not all bacteria have a flagella. But if you can't produce offspring, that's irreducible. Let's see how that works. That little box, we're going to look at that part of the wall of the oviduct. There it is in the microscope. These are tall columnar cells, and up at the top, you see what look like little hairs up there? Those are called cilia. Each of those cells have thousands and thousands and thousands of little cilia. They're so tiny you can't even see one in the light microscope. You say, what do you mean? I'm seeing them up here. You're seeing them collectively. The way you can see hair, you can see that I have at least some hair, but you can't see one hair from where you are. And so these cilia are so small, even under the microscope, we can only see them collectively. Can't quite resolve one all by itself very easily, at least. And these cilia beat. And going down the oviduct, there are just trillions of these cilia. So how do they beat to move the egg? Well, we could say you just sort of fire the pistol and they all start beating. Well, imagine if all those trillions of cilia just started doing this. Would that move the egg down the oviduct? You maybe work up a little foam, that'd be it. I think I'm onto it. It's like window wipers on a car. They go like this, right? All of them together. Would that move the egg? No, the egg would sort of go forward, backward, forward, backward, forward. Oh my, you know what the movement is? It's a metachronal rhythm. Isn't that incredible? You say, what's a metachronal rhythm? Have you been to a ball game where they do the wave? That wave they get going in the stadium, that's a metachronal rhythm. Ever see wind blow across a wheat field? Those waves that go through the wheat? That's a metachronal rhythm, and only that can move the egg in a coordinated way. Let me give you a little example of how these cilia beat. That is a metachronal rhythm. Can you think about this? Can your mind bend that far? Trillions of these, thousands per cell, all interacting collectively down the oviduct in concert, setting up an integrated metachronal rhythm without which the egg wouldn't go down the oviduct, without which there would be no peephole, no mammals. It'd be over. And don't say, hey, look, if you're evolution, we'll get it right next time. We're going to work on this. We'll come back. It's over. This is irreducible. This truly is irreducible. Well, let's look at the whole pattern of reproduction. The egg is released, and when it's first released, you have about a 12 to 24-hour period of time during which fertilization can occur. If it doesn't occur in that window, it's not going to occur. So sperm will be up in this region at the beginning of the oviduct, and if they're in this region at the time the egg is here, there is a good possibility that one of the sperm, at least, will manage to penetrate through and fertilize the egg. And if it does, the fertilized egg will move down the tube. It will go through its first cell division after being fertilized, and here at 30 hours we would have the first two cells. So what was one cell becomes two. Let's look at this fertilization event a little closer. This is a cross-section of the oviduct, cut right across it, and inside, would you believe it, when I cut this section, I happen to hit the egg. To get the picture, you'd have to think of a basketball sort of floating in space someplace in a very long hallway. You make a random cut across the hallway, and you hit the basketball. And so that's what we managed to do. There it is, right in there. It was an incredible stroke of luck, some might say. And around that ball, you see the ball right in the middle, that's the egg, unfertilized. Around that ball is a bunch of messy-looking cells. See all the little blue dots there? They're just cells that accumulate around the egg. There's a very interesting scientific name, these cells. They're called the cells that accumulate around the egg. I'm not going too fast, am I? No. Would we call it the... No, it's the cumulus oophorus, which is the cells that accumulate around the egg. You think, do we need these things? Without these cells, you wouldn't be here. These cells, the cumulus, secrete a sticky stuff. And it's this sticky stuff that the cilia get a hold of to move the egg. They've done experiments where they've stripped these cells off and put an egg into the oviduct, say in rabbits. And the cilia just make the egg go round and round, sort of like a ball on the nose of a seal. You know how they can get it spinning? It doesn't go down the oviduct. Without the sticky goop made by the cumulus cells, it's pretty well over. You won't get a second chance to get it right. You need to get it right the first time. Now let's just magnify that egg cell with its investing cumulus cells. Here it is at higher power, and when we do this, inside of this big cell called the egg, you'll see there's a little blue dot here, and that little blue dot are the chromosomes, or represents the chromosomes of this egg cell, and you can see how much cytoplasm there is compared to the size of all of the chromosomes. And would you believe it, it's like making a section through a long hallway in which a basketball is in three-dimensional space. You not only hit the basketball, but inside the basketball is a little BB, and you hit the BB dead center. Would you mind if I just sort of stood here and gloated over this picture for a while? And those are the chromosomes, because when that egg is ovulated, it goes to form the first, what we call meiotic division. And it waits there with the chromosomes not pulled apart, but the spindle ready to pull them apart, and it won't go any further unless fertilization occurs. Then it'll go on. At 12 to 24 hours, we have fertilization. The fertilization process in itself is just incredible. There could be 100 or more sperm in the vicinity of the egg, and it's essential that only one sperm fertilize the egg. If you get two sperm in there, we call that polyspermy, and there's no possibility of getting a viable offspring here. It'll just spontaneously break down. The sperm cell has three barriers it has to get through to fertilize. First, it has to get through the cumulus. No way you're getting through the cumulus. I mean, you just can't get through. So that's pretty much the end of life right there. Except the sperm makes a highly specific enzyme that digests its way through the sticky stuff of the cumulus. What a stroke of luck, huh, for the evolutionists. Without this, it would have been over. You have to have the specific enzyme get through the cumulus. You're through the cumulus and you're down to the red layer. You see the red layer? That's a shell. You didn't know our human eggs had a shell, did you? Hey, it's not a hard shell like on a chicken. It's a kind of like a skim of jello, you know, a gelatinous, tough, jelly-like shell. And it's called a zona pellucida, which is a fancy name for clear zone. The sperm needs to get through that, except no way the sperm going to go through that, is it going to go through that clear zone. It's a barrier. But there's a second enzyme that specifically digests its way through the zona, without which there'd be no fertilization, be the end of life. Now you're down to the membrane, and remember, we have two cells of completely different sources, one from father, one from the mother. They should have nothing to do with one another. In fact, they should build sort of resistant responses. But they don't. As soon as the first sperm cell, it might be just ever so slightly ahead of the next one, like a horse race, As soon as it touches the membrane of the egg, there's a quick little system that makes sure that it's the same species and all that sort of thing. And if everything is right, the membrane of the sperm and the membrane of the egg open up so that the contents of the sperm goes inside the egg. And that's the first step in fertilization. Remember, you have to keep the second one out. How do you do that? Oh, this is so wonderful that I'm not sure you're going to be able to contain yourself when I tell you this. Just under the surface of the egg cell membrane are a whole bunch of little granules called cortical granules that are right near the membrane. And they're full of an enzyme called ovoperoxidase. And as soon as that sperm touches the egg cell membrane, in just a fraction of a second, the granules explode, send their contents out into the space where the red shell is, and harden the shell. Any sperm not right at the membrane get locked right where they're at. What sort of a creator do we have? What are his limits? Once that happens, once fertilization happens, once the genetic material of the egg and the sperm are together, from that moment on, if you do not interfere, if you let it be, if you let God look on and watch and monitor the process in the womb, if he so wishes to bless you, a baby will be born 9 months later. And I believe it's from this moment on we ought not to interfere. It's the first chance that we, with our limited human minds, can perceive that there's something here that is forming a person known to God because God said he knew him even before the fertilization occurred. He certainly knows him now, or her, and watches on. Psalm 51 is sobering because not only does a person begin then, but like all persons on this earth, the person's a sinner. A fertilized egg is a sinner. Well, how about that for a definition of a person? Look in Psalm 51, verse 5 and 6. Behold, in other words, the psalmist wants to get your attention. This is important. Behold, I was brought forth in iniquity. That means coming out of the womb. I was a sinner. As the baby emerges, he was a sinner. But he goes further than that. And in sin my mother conceived me. That's the fertilization event. That's from the very beginning when life of the individual began and the person began. So we are sinful from the beginning. Behold, you desire truth in the inward parts, that is in the heart. And in the hidden part you will make me know wisdom. Since we are a sinner and we're just a sinner from the beginning, we pray to God as the psalmist did in Psalm 51. To me, verse 10 and 11 here are simply one of the most important verses in the Bible. We spend a lot of time talking about creation. But the creation of heaven and earth is not the really big creation. The creation of heaven and earth, even when God says, oh, and he made the stars also as though it were, oh, there's something else. Oh, yes, all the stars. By the word, by speaking the word, there is a more phenomenal creation. There is a greater creation than that. And here it is. Create in me a clean heart, O God, and renew a steadfast spirit within me. Do not cast me away from your presence. Do not take your Holy Spirit from me. See, when God created the stars, he didn't have to argue with them. They didn't put up a fuss. They didn't say, we'll get together next week, we'll discuss this again. Adam and Eve didn't put up a fuss. He spoke, and it was. But when man fell into sin, he's got this stony heart. The Bible says he's at enmity, he's at warfare with God. And God needs to come in and create a new heart. I call that the second creation. And the third creation is he's gone to prepare a place for us. Well, back to our oviduct. We get out here to 30 hours after ovulation. We now have 2 cells. It's divided. We're on our way. Remember, every time you divide, 2, 4, 8, wow, you pick up cells in a big hurry, don't you, doing that, exponential growth. This is what the 2 cells look like. In a human, the egg is just divided, fertilized egg, and it's the first 2 cells made. And if you look, you can see the shell, the zona pellucida, going around it. Now, when you get into the adult or later in development, the cells are all held together by little buttons called desmosomes that kind of keep them from falling off and getting lost. There are little glues and buttons that hold them all together. But at this stage of development, there's really nothing holding the cells together except the shell. If the shell is defective and breaks down, those two cells can separate. Now, this is so heavy duty, you may want to plug your ears and not hear this because you're probably going to worry about it. But I've learned to just get blown over by God and just say, Lord, you know. I don't have to know. If they separate, you get two people. And if they stay together, you get one person. Each of you in this room could have been two people. Maybe some of you are. You have any identical twins here? Because an identical twin is a virtual clone. I have people say, oh, if they make a clone, would it be a monster? Would it have a soul? I say, you've probably met a clone in your life once or twice. Really? Yeah, an identical twin. It's a clone. But when God makes a clone, it works. You see, because these aren't germ cells anymore. This is not an egg anymore. This is not a sperm. These are body cells, you might say at this point, albeit embryonic ones. And if they split two, if they stay together one, how do we explain that? We don't. We leave it up to God. He knew them from eternity anyway, so. Now, most twins are not this kind. Most twins are where, instead of one egg ovulating, two or more ovulated, and then they got fertilized. Now, there you could have a male and a female, right? But here they're going to have to be the same sex. In fact, they'll have exactly the same genes. You think, man alive, they must move at the same time. No, they each have their own personalities. One could conceivably be saved and one not. In fact, if you want to really think about what little we know about genetics, identical twins have exactly the same DNA as far as we know, but they have different fingerprints. Where are they coming from? I'm mainly here to give you questions, not answers. Well, we move on. About 3 days, you got a ball of cells, which is given the scientific name of